Welcome to Lecture 5 entitled Cell Structure. In this lecture, we're going to be looking at tons of cell biology and figuring out all of the stuff that we've based our knowledge on, how it applies to cell biology, and the way that we look at cells. Before we begin, we want to understand one thing, this idea of microscopy. Microscopy is a way that we can look at cells. But what does it entail? What is microscopy? So microscopy will be our first flowchart. It contains about three major features that you should understand. And we'll write those down right over here. Three features. These three features include the ability to magnify. We'll call this magnification. Magnification. Microscopy focuses on things like resolution, making sure that there is a strong positive resolution in its practice. And lastly, there's contrast involved in microscopy. We'll talk about each of these right now. So what does magnification mean? Very obvious, but speaking in terms of scientific terms, we can say that magnification is defined as the ratio of the size, and let's write this down, the ratio of the size of the image seen, so the image that we're viewing, the image seen in relation to its actual size, so to its actual size. So that's a scientific way of defining magnification. Basically, we're going to be looking at something and we're going to be magnifying it and making sure that that magnification gives us this ratio in which the size of the image, let's say, is 1,000 times, 1,000 to 1, bigger than it is in actuality. That's the basic idea behind magnification. Next, we can look at resolution. Resolution is simply defined in microscopy as the measure of clarity. So we can write this down as our measure of clarity. How clear an image is. What is its resolution? And resolution has a big time sort of emphasis on the idea of focus. Focus basically entails the idea of wavelengths, and we won't get too much into the physics of this, but more so the knot wavelength of radiation is what gives us a good idea of what focus really means in terms of resolution and in terms of microscopy. So wavelength of radiation, think of it like this. If you have a long wavelength, you can have two types of wavelength, a long wavelength or a short wavelength. If you have a long wavelength, I'm going to tell you that you're going to have a bad image. So we're going to squeeze this down right here, a bad image. The image won't come up clear. And then conversely, if you have a short wavelength, all you need to know is that this is when you're going to have a nice, clear image, high resolution. So high resolution is obtained when you have a strong focus. When you have a strong focus is when you have a short wavelength. This gives you a nice measure of clarity, gives you a nice positive resolution. Lastly, contrast refers to making things stand out. Simple as that. So we'll write that down. Contrast in microscopy is all about making things stand out. Oftentimes this is done through a process known as staining. Many scientists in the lab, what they'll do is they'll stain specific parts of a cell, let's say, if we're talking about cell structure in this lecture. Cell structure, if we want to look at a specific organelle, a specific part of the cell, we'll stain it and then look for that contrast in our microscope when we're completing microscopy. The last thing we want to talk about in terms of microscopy are the two major types of microscopes. So we'll write types of microscopy. There are two types that we want to focus on and understand before we get into actual cell structure. There are two types of microscopy. I could actually say that there are two types of, let's say, microscopes instead. Microscopes. One of them is a light microscope, and I'm going to do that one right over here. So light microscope. And the other is known as an electron microscope. We've heard of the term electron before, and we'll see why it's called that in just a second. Electron microscope. So what is a light microscope to begin with? A light microscope What it does is, and this is very simple, simply speaking, it takes visible light, and that visible light is taken from a light source, like let's say a light bulb that's within the light microscope. Visible light from light source. And what does it do with it? What does it do? It passes through the object. So visible light from light source passes through. And we just understand that it's passing through whatever you're trying to magnify, whatever you're trying to look at through the microscope. This passing through causes light to bend, so we'll say it bends light. And when we bend light, this is our key point of emphasis, this is when we get magnification. That's all you need to know. Nothing more than magnification is due to the bending of light coming from the light source of, now you know why it's called, a light microscope. A light microscope often involves staining. We've talked about staining. It helps us visualize or contrast what we really want to see. The amount of magnification on a light microscope is usually about 1,000 times. So you know that ratio. That ratio now is 1,000 to 1. We will see something 1,000 times larger than it actually is, based on that magnification definition. In addition, it will have a resolution, and all you need to know about the resolution is just remember it's the measure of clarity. The units for the resolution in this situation will be 2 micrometers. So micro is usually written down as a U-M. So 2 micrometers of resolution. I'm going to write resolution here. And lastly, in terms of light microscopes, there's one major problem that we can speak about, and I'll just drag that down right over here. The major problem with light microscopes is that there's not much known about the internal structure. It doesn't tell much about internal structure, so we can write that down as not much info. Just rewrite that. Not much info about internal structure. Basically, that means that I can see something from its, let's say, exterior, but I can't really go into its interior and look at it. Why is this relevant to cell structure? Well, we can see cells with light microscopes, but we cannot see the inside of cells with a light microscope. It's simply not strong enough. What is strong enough? Electron microscopes. Electron microscopes are very strong types of microscopes that help us see internal structures. They're called electron microscopes because what we do in this situation is we take and focus a beam of electrons, at the thing that we want to magnify, let's say on specimen. This provides us with a very strong resolution and also a very high level of magnification. Specimen. The magnification level in this is about 250,000 times magnification. So that's our ratio. We see something 250,000 times bigger than it actually is when we look at an electron microscope. There are 2 types of electron microscopes, and we'll briefly go over them right over here. The 2 types are TEM and SEM microscopes. A TEM microscope stands for transmission electron microscope, and SEM stands for scanning electron microscope. Only thing you really want to know about these is that a transmission electron microscope involves putting the specimen in plastic slides, and a scanning electron microscope actually involves putting the specimen, putting a layer of gold film on the specimen, and that gold film will allow electrons to pass through. Just like we stated here, same thing with the transmission, but instead we are passing through a plastic film. So transmission electron microscope, electrons pass through a plastic sort of film, and the scanning electron microscope, it passes through a gold film. One thing I want you to know about the scanning electron microscope, it actually produces a 3D image, a very nice 3D image. You can actually see the 3-dimensional shape and structure of things within a cell, let's say. So the SEM is usually the one that's preferred over the transmission electron microscope, but the electron microscope, just like the light microscope, has some disadvantages. So we'll say some minor problems that are involved with the electron microscope. First and foremost, it actually kills cells. When you're when you're beaming electrons through a specimen, you're going to kill the cells. That's just how the physics and chemistry works. So that's our problem. We can't see things living. We actually can see living, moving cells with a light microscope. So there's that disadvantage advantage. In addition, the electron microscope sometimes will alter the structure. So it alters structure. Because it's such an invasive procedure, invasive basically means that it goes in, actually physically goes in and does something, let's say, negative to the structure. It's a very invasive procedure to beam electrons at something, so that sometimes the structure is altered. In addition, it's often very, very expensive. You will probably not have the ability to experience an electron microscope, but you will most likely, or already probably have, seen a light microscope and how it works. And in addition, it involves lots of preparation. So you get this great, beautiful 250,000 time magnified image. You get this 3D image, but there are these problems that you have to focus on and understand that come with the electron microscope, and you as a scientist have to figure out, will the pros outweigh the cons in terms of a light microscope or electron microscope when you're trying to figure out what you want to do. So overall, what we want to get from this part of the lecture is that microscopy is necessary to see cell structure. The title of this lecture is Cell Structure. Microscopy involves three major things. It involves magnification, resolution, and contrast. And each of those things are sort of highlighted and seen in the types of microscopes that are available to us as scientists. Types of microscopes include a light microscope and an electron microscope. The electron microscope provides us a very strong, very magnified image, but it comes with these problems, whereas a light microscope is a much easier way to magnify things, but doesn't magnify nearly as much.